All right, so here we are back with our discussion of hemoglobin. Now, what we just looked at on the previous slide was our transition from the T state to the R state, focusing in on that kind of central cavity as being something that kind of closed off between our T state and our R state. Now, what's happening is if we look at this model right here, what we're seeing is, and what I want to focus on, is this blue shaded helix, so helix F. Um, when oxygen binds to our Fe ion, which is in the center of our heme ring, or this heme ring is also known as a porphyrin ring, conformational change is going to occur. Now, keep in mind that we've got a histidine residue. The proximal histidine residue is very nicely coordinated with that iron ion. When O2 binds that iron ion, one of the things that happens is our heme kind of goes from this shape to this shape, or from this position to this position. Where my middle fingers are meeting, that's where the iron ion is. And so it goes from this to this, flattens out in one plane. So we're looking at this F helix, this blue F helix. That's with the iron ion outside of the heme plane. Now, when oxygen binds, what happens is that iron ion is pulled downward and put in the same plane as that porphyrin ring. That E, that iron, pulls along that histidine residue in that helix. So it's almost like the domino effect is iron binds to oxygen, but iron is also bound to the histidine. And so as it binds to the oxygen, it pulls the histidine along with it. Now, those are minor differences there. Those are subtle differences there. And what also happens is we have these other interactions, as is displayed. I'd like you to focus in on the bottom half of this figure, um, where we have interactions. For instance, we have a aspartate interacting with a tyrosine. Well, as soon as we transition from that T state to the R state, that aspartate residue, well, here's where it is now, and here's where the tyrosine that it previously interacted with is. These movements are minor movements, but they cause disruption of different types of interaction. So that oxygenation causes the F helix of one of those alpha beta dimers to shift relative to the C termini of the opposite alpha beta dimer. Now, O2 binding causes a conformational change in the subunit. These minor things, O2 binding causes the structure of the protein to change ever so slightly, which as more O2 molecules bind, hemoglobin transitions from that low affinity T or deoxy state to that high affinity R or fully oxygenated state. So what we're doing is we're seeing what's known as cooperative binding. The observed oxygen binding has a sigmoidal curve. Now, so this can only be seen in multi-subunit proteins because we're going to see one subunit impact the other subunits, which impact the other subunits and basically increase their affinity for binding of O2. So oxygen binding changes myoglobin and hemoglobin conformation. But the important thing is that myoglobin, if you remember, is a single subunit. Hemoglobin, whenever its shape changes, whenever one of those hemoglobin subunits binds to O2, that subunit changes shape, which causes adjacent subunits to change shape, which impacts their affinity for oxygen. It increases their affinity for oxygen. This is referred to or known as positive cooperativity. If you look here, what we're seeing is a model representation of it where hemoglobin at the top is deoxygenated. Whenever it binds O2, well, now all of a sudden the other subunits of hemoglobin want to bind O2. I always kind of think about hemoglobin, or I always think of positive cooperativity as if you ever been to a wedding where there's dancing, and maybe a wedding's not the best option, but like when you're in junior high school, if you ever went to a dance, I never went to the dances in junior high school. Um, Maybe it's because I would say junior high school. At any rate, nobody wanted to be the first person to dance. I went to a dance when I was in sixth grade, but I was actually in seventh grade. it was a sixth, anyway, my sixth grade class, we got a pizza party because we did the best in the accelerated reader, and it was a pizza party / dance. But they had to wait till the next year, so we were all in seventh grade, but it was at the sixth grade campus. At any rate, nobody wanted to be the first person to dance. But then somebody finally broke the seal and danced. And then all of a sudden, everyone else saw that and they're like, hey, that's a good idea. I want to dance. And so binding to the first O2 molecule, nobody wants to do that. No subunit wants to do that. But as soon as one subunit does it, then the other subunits say, oh, well, I guess I can bind to O2. And by the time that we're approaching being fully saturated, now every subunit is like, oh, well, I always wanted to bind O2, much like at the middle school dance. Everyone's like, oh, well, I just had so much fun dancing. It was my plan all along. They're acting as revisionist historians there, but nevertheless. Okay, so just as much as we want O2 to bind or hemoglobin to bind O2, we also want hemoglobin to release O2. And so we have this graph that looks like this. Our theta or our Y percent saturation is displayed on our Y-axis, and our L concentration or PO2 is displayed on the X-axis. Hemoglobin's oxygen binding curve is a sigmoidal, not hyperbolic shape, so we need to modify that equation to display as much. And we display it using the Hill equation. The Hill equation shows these numbers of O2 displayed as n and therefore displayed as exponents here. So the equation is basically the same, but we have that n as the exponent. N is called the Hill constant or Hill coefficient. If n equals 1, you have non-cooperative binding, therefore you have hyperbolic curve. So if you were to take myoglobin and use this exact same equation with substituting n in for 1 or 1 in for n, you'd get that nice hyperbolic curve that we've already seen. If n is greater than 1, you have positive cooperativity, therefore a sigmoidal curve. If n is less than 1, you still have positive cooperativity, or sorry, you have negatively cooperative system, but you still have a sigmoidal shape to your curve. Now, the Hill equation is cumbersome to work with, and all that is needed is n and p50 from that Hill plot. I'm going to be focusing quite a bit more on the oxygen binding curves that we've previously seen. I just want to expose you to the Hill coefficient, to the Hill equation and the Hill graph, what that looks like, so that you have a sense of what it is. So it's not just some radical idea if you've never heard of. Now, here is a Hill plot. I've always kind of thought that the Hill plot was just a nice way to take something that's nonlinear and make it linear. If you take log of y over 1 minus y, you get n log PO2 minus n log P50. You plot the concentration, or you plot y over 1 minus y, log of y over 1 minus y versus PO2. So a log of PO2. So you're plotting the log of your percent saturation over 1 minus percent saturation against, on your x-axis, the log of PO2. So where do we get n and P50 from? When log of y over 1 minus y is 0, or y equals 0.5, then n log PO2 is equal to n log P50, which is kind of similar to when we talk about that same equation without the log representation, where we say, well, P50 is equal to 1/2. Another way of saying this is log PO2 is equal to log P50 when y equals 0.5. N is going to be the slope of the line for each of these different lines. Now, how are N and P50 determined? Well, if you look at the central point when log of PO2 is equal to log of theta over 1 minus theta, when that is equal to 0, and we have a line that intersects with that, that's where our log of P50 is. For the first because keep in mind, we've got hemoglobin that binds 4O2. Log of P50 for the first O2 is displayed right here. Log of P50 for your second and third O2s is displayed there. And the third one, or the fourth one, I'm sorry, is going to be right there. Now, the significance of this and what I want you to take away from this right here is this gives a measure of affinity for the different O2 binding sites. Log of P50 for your first O2 is the weakest. So the lowest affinity is for that first O2 molecule. Your fourth P50 is going to indicate you have the strongest affinity for that fourth and final O2 molecule. Now that's really kind of where I want to proceed with the Hill equation and the Hill plot, because I'm most interested in those nonlinear, those sigmoidal curves. Now, what about the pressure of O2 and the P50? Well, the PO2 for blood is going to vary, whether it's arterial or venous blood. Venous blood, the PO2 is approximately 30 torr. Arterial blood, it's 100 torr. So the pressure of oxygen is much higher in the arterial blood than it is in the venous blood. Myoglobin shows no cooperativity. P50 is equal to 2.8 torr. So one way to think about that is as long as the O2 concentration is greater than 2.8 torr, myoglobin is going to be at least 50% saturated. Hemoglobin, on the other hand, displays cooperativity. Its overall P50 is about 26 torr. So thinking about venous blood, well, venous blood, most hemoglobin is going to be 50% saturated. Hemoglobin only has two quaternary forms, the T shape, that deoxygenated form, and the R shape, that oxygenated form. The T to R transition must occur simultaneously at the interface between the two alpha beta protomers. Now, the first O2 binding is going to be where you have very low affinity. You're dealing with that T shape or that T structure. You see no cooperativity with a P50 of 30 torr. Now the transition from T to R is with our second O2 binding. The interface is destabilized enough for the T to R transition to occur when we bind that second O2 molecule. Our third O2 molecule is going to be a little bit easier to bind to because now we're in that R state that has a slightly higher affinity. So what we see ultimately is our fourth O2 binding occurs very readily with our high affinity R state. We see no cooperativity and our P50 is 0.3 torr. So what we're seeing here is that P50 is decreasing ever so much. First O2 required 30 torr to be 50% saturated. The third is about 12 and the fourth is about 0.3 torr. So what this means is for that fourth O2 to be 50% saturated or present, it doesn't require a lot of O2 because we have a very high affinity for that. Now, how does this cooperative binding impact what we want to do? We want to bind O2 and release O2. So inspired air, we have a concentration of approximately 158 torr, considerably higher than all of the P50s that we looked at. Alveolar air, arterial air, well, binding is going to occur in the alveoli within the lungs. The pressure of O2 is about 100 torr. So we've got plenty of oxygen there to saturate. Meanwhile, in the capillaries, our O2 concentration drops considerably. In the interstitial space, it drops even more. In the cytosol of a cell, it's about 10 torr. So our O2 concentration drops a whole lot. And so that's where we're going to see that release. We're going to see release of our O2 when we're talking about or when we're in the capillaries, and then ultimately it's going to be completely released or pretty much completely released when we're in the cytosol. So cooperativity is designed to release O2 at very low pressures. Those pressures are generally going to be lower to or comparable to our P50s. So one way to look at this, and this is just a nice little demonstration, that our T state, it's like a teeter-totter. Our R state is like a teeter-totter that's shifted in the other direction. So what causes that transition? The binding of oxygen. An oxygen is going to bind in the T state. A second one is going to get us closer to the R state. A third one is going to get us to the oxygen in R state, where we then have the highest affinity for our last and final O2 molecule. Now, these different shapes, the T shape and the R state, there are tons of proteins that have T versions and R versions. There are different proteins that, and it's not strictly oxygen binding proteins, but the term allosteric is really used to give information about a protein with two different shapes. An allosteric protein has multiple shapes. So hemoglobin is an example of an allosteric protein. Now, binding of an allosteric effector at one site affects the binding properties at another site. The conformational change induced by the effectors interconvert a more active or a less active form of the protein. So converting between the R and the T is going to, and the T and the R is what's going to be taking place. Hemoglobin is a great model for protein ligand binding, just as myoglobin is a great model for protein ligand binding, but hemoglobin is also a great example to illustrate allosteric. The structure of oxygenated hemoglobin or oxyhemoglobin is different, as we saw here. This is our T state and our R state. Now, to the layperson's eye, they look very similar, but they are different. Oxygen is an allosteric effector. So going back up here, the conformational change induced by the effectors interconvert the R or T forms of the protein. So oxygen is an allosteric effector as well as a ligand. So it both is the natural ligand that hemoglobin wants to bind to, but it also induces these changes. Now, that's not true of every single molecule that hemoglobin is capable of binding, but it is an example of what is known as a homotropic interaction. The ligand and the effector are identical. What this means is that hemoglobin can bind some molecules, and it's not going to change shape. And those molecules are not necessarily a natural ligand of hemoglobin. So a heterotropic interaction is when a molecule or when a protein binds a molecule, its natural ligand, and the shape of the protein does not change. So I think the best example of an allosteric effector would be a homotropic interaction or heterotropic interaction. So hemoglobin binds a protein, as is displayed here. Our T state will bind to this blue square, this allosteric effector, and then oxygen comes around to bind, but that allosteric effector says, I don't care. And only whenever it gets enough of that oxygen is that allosteric effector basically competed away. So an allosteric effector or an allosteric kind of inhibitor can basically block and keep a protein in the T state. Now, here what we see is the sort of curve that I want you to be comfortable with and I want you to be familiar with. The results of the binding of an allosteric effector may be seen or will be seen on the oxygen dissociation curves. So I'm going to say plus EFF effector plus other effector. So what an allosteric effector can do, or what an example of an allosteric effector is basically anything that shifts the curve to the right. And whenever this curve shifts to the right, that is an illustration of a substance that causes what's the term that I'm looking for? What's the word that I'm looking for? That shift to the right is going to cause the P50 to go up. So that's going to cause your protein's affinity for O2 to go down. And we see that with these particular examples. So similarly, an allosteric effector can cause the affinity or the curve to go to the left, which causes the P50 to go down, which means the affinity goes up. Now, there was a researcher by the name of Christian Bohr, or a scientist by the name of Christian Bohr, and he studied pH. He studied how pH impacts O2 binding. And for this graph, what I'd like you to focus in on is I'd like you to focus on this curve right here. We've got an oxygen binding curve, and the environment has a pH of 7.2. What Christian Bohr did was he said, and I'm going to scribble out the myoglobin, I don't care about that. What Christian Bohr did was he said, all right, well, I'm studying hemoglobin, and I'm going to study its ability to bind O2 if the pH is 6.8, 7.2, and 7.6. So he said, all right, well, if we operate under the idea that 7.2 is kind of our physiological pH or mid-range pH, let's see what happens when the pH goes up. Let's see what happens when the pH goes down. So if the pH goes up, our hydrogen ion or our proton concentration decreases. And then if it goes down, our hydrogen ion concentration increases. And an important thing to remember, if the hydrogen ion concentration goes down, that means some of the groups, some of the proteins that we're dealing with, maybe they've become deprotonated. Similarly, or kind of conversely, if the pH goes down, then some groups are probably protonated where they otherwise would not be. So this is termed and known as the Bohr effect. Binding of protons diminishes oxygen binding, okay? So what we see is if we look at the 7.2 as mid-range, here's our P50. I'm going to draw another line to indicate P50 at 7.6. And then another one, 6.8. Okay, so pH is 6.8. Our P50 is about, let's see, I'm going to call that 50 just for simplicity, 50 torr. At 7.2, our P50 is approximately, I'm going to call that 35 torr. And then at 7.6, I'm going to call that 20 torr. So what we see is as the pH of the environment goes down, our P50 increases. So our affinity for O2 decreases. Then, as we see the pH go up to 7.6, we see that our affinity for O2, or sorry, our P50 goes down. Therefore, our affinity for O2 goes up. So essentially what we're looking at is there's kind of a relationship between affinity for O2 and the pH. which is interesting and worthwhile to note because why is that happening? Because clearly, if there's protons in that environment, something's happening to our protein. And so what Christian Bohr did was he said, all right, well, let's get to the bottom of this. The pKa's of ionizable groups involved in the favorable ionic interactions are perturbed, they're changed. So our deoxyhemoglobin, our T-state protein, there are favorable ionic interactions or salt bridges that stabilize the structure, that stabilize that T-state. Specifically, the protonated form of histidine 146 at the C termini of the beta 2 subunit forms an ionic interaction with aspartate 94. So we have an ionic interaction between histidine and aspartate. The ionic interaction stabilizes the proton against dissociation, increasing the pKa of His 146. Okay, so the ionic interaction stabilizes the proton against dissociation. So this is kind of it's almost circular logic. Because there is this ionic interaction, this favorable ionic interaction, there will always be a favorable ionic interaction because the pKa is going to go up because that's going to remain protonated. Now, oxyhemoglobin, on the other hand, upon binding of O2, the favorable ionic interactions are disrupted. Histidine 146 aspartate 94 interaction is disrupted, returning the pKa to their normal values. Histidine 146 is deprotonated in oxyhemoglobin at a normal blood pH of 7.4. So, not to say that it was just the silver bullet, but these two amino acids, His 146 and aspartate 94, were pretty well studied. And that's what Christian Bohr dove into a little bit deeper. He said, okay, well, we've got these interactions between these two amino acids. We've got this ionic interaction that's stabilized. And so it seems as if that has some impact on ultimately the protein's ability to bind O2. So the high hydrogen ion concentration, which is the same as the low pH, favors the protonation of histidine 146, which stabilizes our T state. The T state favors the release of O2, which is analogous to saying it favors or it results in a higher P50. Anything ultimately that favors the T state lowers O2 affinity, causes the P50 to go up. Histidine 146 is not the only residue responsible for the Bohr effect. The mechanism is similar for a number of other residues. So ultimately, the interactions between different residues and different subunits are going to be what stabilize this protein structure. And anything that favors the T state forming is going to make it harder to transition from the T state to the R state. Therefore, your P50 is going to go up. So there's tons of relationships between P50 and stabilization of a structure. So if you could add something to hemoglobin that stabilizes the R state, what is that going to do? Well, it's going to cause the P50 to go down. And when that P50 goes down, what does that mean, or what are the implications of that? That means your affinity for O2 is going to go up. All right, well, I hope this was helpful, and I will see you in the next lecture. All right, everyone, have a good day.